Welcome everyone. Thank you for joining our webinar. We're going to wait till about 3:05 to get started to give the students some time to log on. How's everyone's week going? Olivia, do you have any plans for Thanksgiving?
No, I'm staying here, staying inside.
Probably the safe bet. What about you, Lacey?
I'm driving back to California on Saturday. So I'll be here for Thanksgiving though. Yeah.
Are you going to spend it with friends? Any friends?
My sister lives here, so I'll be with her, and then my parents and my brother are coming up, so I'm excited.
Nice. Anybody else have anything fun they're doing for break? Anything to relax?
My family is still doing a nice little socially distanced family dinner on Thanksgiving. My grandma really wanted to this year.
Yeah, I found that my grandma's been the hardest one to convince about the whole social distancing thing, but she's in California, so I won't be seeing her this year. Chris, do you have any plans?
I'm just hanging out here in Corvallis. Nothing too exciting.
All right, it's 3:05. I think we'll get started if everyone's all right with that. So in case if you would like to follow along with our activity, these are the supplies that you will want nearby. So you'll want 3 long bendy items. I'll be using string. You could also use pipe cleaners or even shoelaces if you don't have string. You'll also want about 10 cotton swabs or toothpicks if you don't have Q-tips. In addition, you'll want some tape and 4 different colors of markers, preferably red, blue, black, and green. You'll also want scissors and a pair or something to write with, as well as a piece of paper to follow along with our activity. Now for some just quick introductions. My name is Maya, and I'm a second-year student at Oregon State. I'm majoring in biochemistry and molecular biology. And one fun thing I like to do outside of school is I like to read.
My name's Olivia. I am also a second-year student studying biochem at OSU. And outside of school, I enjoy running and yoga.
I'm Lacey. I'm a first year studying biochemistry, and outside of school, I like to draw and paint.
My name is Chris. I am a third year at Oregon State, majoring in biochemistry and molecular biology. And I just like to work outside of school, involved in some research labs, do some occasional coding.
I'm Nicole. I am also a second year studying biochemistry and molecular biology. And one thing that I like to do outside of school is ride 4-wheelers.
Great, thank you all for those introductions. So again, welcome and thank you for joining us for our webinar about DNA. We're excited to have you with us today. If you have any questions throughout this webinar, feel free to drop them in the chat and one of us will respond to them. Or if you have questions after the presentation, you can email them to precollege programs at oregonstate.edu, and I'll drop that email in on the last slide just so you have that. So as we all have said, we're all biochemistry and molecular biology majors, and so we're all part of the biochemistry club at Oregon State University. And in case if you're wondering what biochemistry is, it's the science that studies the chemical processes that occur in living things. And so because we're all majoring in biochemistry, we're learning a lot about biology and chemistry in school. And so we wanted to teach you all about one important topic in biology, and that is DNA. DNA is considered to be the basis of all life on Earth, and by the end of today, we hope you learn at least one new thing about DNA.
So like Maya mentioned, DNA is the basis of all life, and it is what makes you so uniquely you. Take a minute to think about what you already know about DNA. I'm going to write down a couple things too. All right, you can keep writing if you want, but you may be surprised about how much you already know about DNA, but there's endless amounts of information to learn. The basic structure of DNA is a double helix, which you can see on this slide, and we will learn more about later. DNA is located in the nucleus, and it is the basis of genetic information. It serves as the hereditary material for organisms, meaning the DNA is how you receive traits that are so similar to your parents. Now let's take a closer look at DNA. Before we get into the structure and function of DNA, it's important to review a brief history. Rosalind Franklin was a female scientist who took some of the first images of the structure of DNA. She used a fancy technique called X-ray crystallography to generate her DNA pictures, which are shown on this slide next to a picture of her. Two other scientists, Watson and Crick, used Franklin's images to further prove their proposed structure of DNA. Lots of other people were involved in the discovery of DNA, but those mentioned are among the most prominent.
So every one of the trillions of cells in your body needs to have its own copy of DNA. And as we mentioned earlier, DNA lives in this special compartment in the cell called the nucleus.
Now, how much DNA do we actually have? If we break it down, we have about 2 meters of DNA in a cell, which is about the height of a standard door in your house, so it's about 6 feet tall. And we have 40 trillion cells in our body. If we do some basic arithmetic with 40 trillion cells multiplied by 2 meters of DNA, that means we have 80 trillion meters of DNA in us right now. To put that in a scale that I think we'll all get, it takes about 150 million kilometers to go to the Earth, to go from the Earth to the Sun and back, Earth to the Sun, which means that with the amount of DNA in your body right now, you can go to the Sun and back about 200 times, which is a crazy number.
So we have a lot of DNA, and for all of that DNA to fit inside your cells, it has to be packed in really tight. And the easiest way for it to do that is to just twist around itself lots of times. This structure is called a double helix because there are two lines of DNA that are connected to each other, and both of them stay spun around into a helix together.
So we're going to zoom in on DNA a little more closely to look at its structure. So recall the double helix that you all just saw. If we were to untwist this DNA to unwind the double helix, our strand of DNA would kind of look like a ladder. The length of the ladder can be thought of as the backbone of DNA. And the rungs on the ladder are our DNA building blocks. These are represented by 4 different letters, A, T, C, and G. For now, just know that there are only 4 different letters. We'll talk about what these letters mean briefly. But for now, know that it's just A, T, C, and G. A stands for adenosine, T for thymine, C for cytosine, and G for guanine. But they're commonly just called A, T, C, and G.
Now, as I mentioned earlier, we have a lot of DNA, and all that DNA is only composed of those 4 building blocks. And if you look here on the slide, it says that A always pairs with T and C always pairs with G. And everyone on Earth is different from each other, and the only thing that's different is the way that they're ordered in our genome. Now, looking at the structure of DNA, we see that a 1-ring molecule is paired with a 2-ring molecule. I'm going to pick on the top rung with adenine and thymine. And this is important because if you had a 1-ring molecule paired with a 1-ring molecule, or a 2-ring molecule paired with a 2-ring molecule, DNA wouldn't have the uniform structure that's seen in that picture. And this structure, the uniform structure, is important because it's easy for repair and replication, which will be touched on in the second activity of this webinar.
So just quickly before we dive into activity 1, a reminder that if you have any questions, make sure you drop them in the chat and one of us will respond to them.
All right, so now we are going to build our very own DNA molecules. Let me just tilt my camera. My microphone seems to be a little odd when I do this, so let me know if it's not picking up on what I'm saying. So as we said earlier, you're going to need, for this activity, you're just going to need 2 pieces of string, or I have shoelaces here, and you're going to need about 5 cotton swabs, and then you're going to need your 4 colors of markers. So we have black, blue, green, and red, and a bit of tape. For right now. We're going to start by making the ladder structure that we just saw in a couple slides ago. So you're going to take your string and lay them side by side. like this. This is going to be our double helix. And then we're going to take our cotton swabs and we're going to lay them in between the strings like this. So we can form that little ladder structure that we were seeing earlier. And now we're going to tape our cotton swabs to the string. This can take a little bit of time, so it's okay if you feel a little behind. If you're watching the recording of this, feel free to pause at any time and catch up. Just gonna tape them together. So while I'm doing this, do we want to talk about
I don't think we quite heard your question, Olivia. Do you want to repeat that? I think you might need to face the microphone better.
While I'm doing this, because this is going to take a little while, do we want to talk about some of our favorite classes?
I don't think I think you cut off again.
Maya, what's your favorite class at OSU?
What's my favorite class at OSU? I think I didn't take this class this year, but last year my favorite course at OSU, it was my intro to biology class. I just really enjoyed how it was such a huge class, and so it kind of like was just a really amazing thing to have so many people learning biology together, I think, and just like seeing the magnitude of it all. I had like a 500 student class, and so we had this really cool classroom, all sat in a circle, and just how the teacher like presented the information and how they had to convey it to like so many students at once. I think it was just very cool how they did the class. Lacey, what has been your favorite class at OSU?
So I think chemistry. I've only had a few so far because I just got here, but I always liked chemistry in high school, and I've just always found that really interesting. So that's probably my favorite right now. What about you, Nicole? I'm gonna hand it off to you.
Mine would have to be a course that I took this summer called Biomedical Ethics. It was really cool to talk about those kind of really theoretical ideas of how we look at medicine and how we apply it, and whether or not certain things are good or bad, and whether or not that's something that we can all agree on. What about you, Chris?
So just about a year ago, I took an upper division molecular biology class. I got into in-depth into some cellular functions. They talked about all different kinds of microscopes, and I thought that was pretty cool.
Yeah,
Great.
OSU has a lot of great science classes. Olivia, do you have a favorite class at OSU?
Very topical, but my favorite class I've taken was the 311 genetics class because I really like DNA and I think it's really cool. It does a lot of cool stuff. And by now, perhaps we have finished taping our structure together. So this is our basic ladder that we have that we were talking about. And now we are going to take our colors. And what we're going to do is we're going to color just one side of each rung. So you're going to take a color, any color really, and you're going to color a little section . So as you can see, I've colored just a little section on one side, and it's important because we're going to have to fit two colored sections on each rung of our ladder. So then we're going to go ahead and color all the rest of them. And it can be any combination of colors you want because we have any combination of our 4 base pairs in our DNA. But we're only going to color one side of the rung for now. And it can be a little fiddly, so it's okay if you don't get the whole thing completely colored. It can take a little bit of time to color Q-tips. All right. So it's okay if you haven't finished coloring all of one side yet, but hopefully you got a couple rungs done. Maya, could you put the slides back up for just a second so we can see the table? So we have the 4 colors we just talked about. So blue is G, and green is C, A is red, and black is T. And as we were talking about earlier, those certain ones need to pair together. So we're going to put blue with green, red with black. And so when you're coloring the other section of your DNA rung, you're going to do it with the corresponding color. So we're going to do, so I colored this first one blue, which means I have to color the other one green to represent the C and the G. In the second rung, I've colored black, so I need to do the corresponding color, which is red. And so while we're doing this, can you hear me? While we're doing this, do you guys want to talk about your favorite parts of campus?
Sure, I'll start. So I think my favorite part about OSU, in addition to I think all the classes and just having all the students on campus in a normal year, I think one of my favorite things is probably the opportunities. I think I don't know if that it directly answers your question, Olivia, but I think one of my favorite things about campus are all the opportunities and all the different things get involved with, whether that's volunteering or intramural sports or different clubs or activities. I think it's just, yeah, a really amazing campus for that. What about you, Lacey?
So since everything is online, I admittedly haven't been out and about a lot, but I think my favorite is like you can walk out to the agricultural fields and see like the cows and llamas and stuff. So that's probably my favorite part.
One of my favorite parts of campus, I would say, is just the big quad out in front of the library. I love to go up to the upper stories of the library and find one of the desks next to the windows and look out over campus. It's a really pretty campus, but the tricky thing about the library is the higher up you get, the better the view, but you have to be even quieter.
I think my favorite part of campus is in the spring, all the trees will flower and it's really pretty. So perhaps we've finished coloring our DNA ladder, and if you haven't finished, that's okay. But what we're going to do now is we're going to take our strings, and we're going to twist it so that we can form our double helix. And then, look, you made DNA. You have made a DNA model. I think we've all made some, and we're going to show them, right? Right. And that is making a DNA molecule.
Thank you, Olivia, for leading us through that activity. Now that we all have made some DNA, let's think a little bit more about how DNA is multiplied. So each day our body is busy growing and repairing itself. Cells in our body must multiply to allow this growth and repair. Every one of these cells, as we said before, must contain a copy of DNA because DNA is our genetic code. It tells our body how to function properly. And so because every cell in our body needs to have DNA, DNA must multiply when our cells multiply. And it does this by basically just making a copy of itself. But how does DNA copy itself? Well, DNA starts in the double helix that you all just built. Then you can unwind it and make it kind of flat, just how you can unwind your own DNA molecule that you made. Then it can unzip, kind of like a zipper on your jacket, which is separating the rungs on a ladder, separating the base pairs. At this point, the DNA is unwound and is able to copy itself. DNA is able to copy itself by using 1/2 of the ladder as a template, upon which new A, T, C, and G base pairs can attach. These new base pairs connect to the old rungs on the ladder to create a whole new ladder. And this new ladder is a new DNA strand. If this is still a little confusing, don't worry. We're going to walk you through exactly how this works in our next activity.
All right, so we're gonna do activity number 2 now. So you're gonna wanna take some scissors, or if you have an adult with you, they might wanna do this part too. So we're gonna cut up through the middle of our DNA molecule. So I'm gonna show mine. Everyone can see that. So you're gonna cut straight through, and I think if we go back to the slide, if you follow the red line, so if you want to, you can cut it, or actually if you're using Q-tips, if you kind of bend a little bit, the Q-tip will usually break in half. So take a second to do all of your rungs, and then the second picture on the slide, if Maya can put it back up, should be what it looks like when you're finished going through each Q-tip. So I think we'll wait a couple more minutes or seconds, because if you're bending them, it might take a little bit longer, but if you want to look up here for reference, once you're done, you should have something that looks like this. So you'll have 2 half strands. I'm gonna go ahead and cut mine in half because I did a little bit more. All right, so once you're done with that, if you got to 5 rungs, that's fine. If you did more or less, that's okay too. But now we're gonna copy our DNA. So you're gonna wanna get however many rungs you had before, get equal number of Q-tips, and are gonna cut or bend those in half, whichever you wanna do. You're just going to need enough halves until you have an equal number to the rung. So here I cut until I add 5 halves. So I'm going to go ahead and take that away. So now we're going to go back to our base pairing rules, which were put in the chat earlier, but I'll just say them again. So since I have a blue rung originally, that was going to pair with green. So I'm going to color this Q-tip half green. And then I'm going to do that for each color on my ladder. So if you need to go ahead and look in the chat to see what colors we're supposed to go with what colors, go ahead and do that and take a second and color all your Q-tips. So just as a reminder, black goes with red. So now I'm coloring those to match mine. And everyone's will be different. So don't worry if yours doesn't look like mine. All right, so now that we have all of them colored, we're gonna copy our DNA. So we're gonna take some tape and basically make a new rung of our ladder. So take a piece of tape, and this part might be a little tricky, so don't worry if it takes you a couple seconds. Put the tape there, and we're gonna add our second part. Kind of just wrap the tape until you get something that looks like this, and do that for each new pair. So you should have 5 Q-tips once you're done. Oh, and if you mess up, that's okay. I just messed up my base pairs, so that's okay. If you need to get a new Q-tip and draw it again, that's fine.
There's a question in the chat that asks what happens when base pairs are mismatched in the cell. And there's an enzyme or another little protein in the cell that'll run through and double check everything. And if it finds that there's an incorrect base pair, it'll replace it with the correct pair.
All right, so if you look up here, I have 5 new rungs in my ladder. So my DNA has been copied. And now to complete our new strand, we're going to take another piece of string, cut it about the same length as your original piece. And then this is kind of like the first step of the activity, so like Olivia did, but we're just going to tape each Q-tip back onto the string to complete our new DNA strand. All right, so once you do that, you should have a new ladder and your copied DNA. So once you have that, you can twist it again. And if everyone wants to show theirs, there we go.
Thank you, Lacey, for leading us through that activity. Now that we know a little more about how DNA is copied, take about 30 seconds to write, what do you think might happen if DNA is not copied correctly? So think about this question. Feel free to drop your responses in the chat or write it down on your own piece of paper. But yeah, think about the question, what would happen if DNA is not copied correctly? I'll write down what I think too, but take about 30 seconds. All right, so the first thing that I would think of is that if DNA is not copied correctly, it could cause something called a mutation, which is like a permanent change in your DNA sequence. This is a permanent change in your genetic code. These errors in your genetic code can harm your body by leading to diseases such as cancer. But thankfully for us, our body is very good at catching any mistakes, and so our genetic code is generally without errors. It's very, very rare that these mutations will go unnoticed. At this moment, remember that if you have any questions about what we've talked about up to this point, to drop your questions in the chat.
So if you found what we've done here today in this DNA activity interesting, you may have fun pursuing a career that incorporates biology and chemistry into your work. Forensics is a branch of work that relates science to crime and law. And forensic scientists even use DNA to find criminals. Like we've learned here today, the structure of DNA contains base pairs like rungs on a ladder, and everyone on Earth has a different sequence of base pairs that make up their DNA. When forensic scientists go to a crime scene, they can match spit, hair, and blood to DNA sequences in order to catch a criminal. Working in genetics and biochemistry is a great way to further study DNA and how life works.
So to sum everything up, DNA is a double helix and it has 4 bases that we call A, T, C, and G. These bases pair up so that A and T fit together and C and G fit together. DNA is also able to make copies of itself to be the blueprint for new cells.
Well, thank you all for joining us today for this webinar about DNA. We really enjoyed teaching you all a little bit about what we've learned at Oregon State University. Again, if you have any questions, feel free to drop them in the chat or email them to us at precollege@oregonstate.edu. I'll show that email again in a minute, but again, just thank you so much for joining us. We really had a lot of fun today. And I hope you have a great rest of your day. And here is the email if you want to write it down. And if you have any questions, we'll stick around for a few minutes to answer them. And just in case if you're interested in learning more about DNA later, here are some of the sources we used for our presentation.